Good morning, everyone. Today we'll be talking about Chapter 3, the cellular levels of organization. In Chapter 1, we talked about basic anatomy and physiology concepts, talked about things like planes and orientations, and in Chapter 2, we talked about atomic structure, how those atoms are able to form bonds, to form compounds, and then we moved into talking about some macromolecules, things like lipids, which we also call fats, carbohydrates / sugars, proteins, and DNA and RNA. So we'll talk about how those macromolecules kind of come together to form the more complex structures of the cell today. So if you remember in that first class session of lecture, there was that figure that showed kind of the different levels of organization. And so we're kind of slowly but surely working our way through those. So in terms of what's expected of you this week, please make sure that you answer the study guide questions for Chapter 3. Come ready to talk about those on Tuesday and Thursday. For next Friday, make sure that you have quiz 3 for Chapter 3 completed, and then kind of not this Monday, but the following Monday. I guess it depends on when you watch this as to what that means, but just make sure the week that we start Chapter 4, that you take homework for the adaptive practice for Chapter 4 by that Monday. So today we'll talk about some of the three main parts of the cell, talk primarily about the plasma membrane, some of the functions of the plasma membrane, to kind of discern and differentiate between the cytoplasm and the cytosol, and then talk about the selective permeability of the plasma membrane, the electrochemical gradient, and some of its components, and then the processes that transport substances across the plasma membrane. And then we'll kind of wrap up, again, kind of concluding the different components of the cell. So this introduction is very similar to what we saw with the study guide, the different parts of the cell, the importance of the plasma membrane, the components of the cytoplasm. I don't think we'll get to talking about mitosis and meiosis in this particular lecture, and then understand the effects that aging has on the cell. Okay, so we're going to start with talking about the different parts of the cell. So the next figure I'm going to show you, there are going to be a lot of labels. I'm going to circle each of the kind of main parts of the cell that we'll be talking about, and we won't be going through every single labeled component of this cell. Okay, so the structures of the cell. Let's go back to the structures of the cell. And so for the structures of the cell, it's important that we kind of start, I guess, where our discussion today will start, which is with the plasma membrane. So if we look at the plasma membrane, it's basically just kind of everything that is encompassing the outside of the cell here. So you can see I'm kind of tracing around the plasma membrane of the cell. Now the plasma membrane is a double layered structure. I'm just drawing an arrow to it. Again, it's just kind of encompassing everything within the cell. It kind of plays the role that the integumentary system plays for the body for the cell. It kind of holds everything inside the cell and kind of mediates the exchange of things into and out of the cell. And since cells are, single-celled structures, that interplay of things that flow into and out of the cell are very important. We're going to talk about that a lot today. We're going to talk about what's embedded in the plasma membrane that allows that movement into and out of the cell, and kind of why the cell even utilizes this movement. So the plasma membrane, like I said, is kind of the first big player that we're going to talk about in this discussion. We'll mention quickly the cytoplasm. The cytoplasm is just basically, this gelatinous substance that's located within the cell. It's kind of hard to label because it's just kind of everywhere that there's not an organelle. And when I say the word organelle, I'm talking about kind of the larger structures that I'll be mentioning throughout today, things like the nucleus. Also, let's see, the smooth endoplasmic reticulum would be another example of an organelle. The rough endoplasmic reticulum would be an example of an organelle. And each one of these organelles are kind of substructures within the cell that allow them to carry out a specific function. And so each one of these organelles has a very specific function to do for the cell. If you want to kind of think of the cell as a little protein making factory, that's probably one of the easiest ways to think about the cell, because it's making proteins. It's really what its purpose is to make proteins and interact with its environment, and how it utilizes those proteins to do so kind of varies. So if we are talking about the cell as a protein making factory, kind of the place where the blueprints or the ideas or all of the plans kind of get brought down are from the nucleus. The nucleus contains the DNA, or deoxyribonucleic acid. We talked about that in chapter 2. This DNA is located in the nucleus, and that gets read from DNA to RNA. We talked about RNA last time as well. DNA, if you remember, is a double-stranded structure. RNA can be double or single-stranded, but in this case, we're talking about something that's single-stranded. So DNA makes RNA, makes protein. So this is just kind of the very simplified, set of steps that takes place in the cell. You've got DNA hanging out in the nucleus, and then it gets made into RNA. RNA gets exported from the nucleus or sent out of the nucleus, and then other structures in the cell help it get made into protein. So when I say it gets made into protein, this DNA is getting read, and kind of the last step here is the fully made protein structure. So what allows the cell to actually carry out this function? Well, as mRNA gets exported, that's maybe not the best color. As mRNA gets exported from the cell, as you can see right here, it's going to go and interact with the rough endoplasmic reticulum. And the rough endoplasmic reticulum has ribosomes. So these ribosomes are protein-making machines. So those ribosomes are what allows the cell to actually make the proteins. From there, the proteins get shuttled to kind of this little globular structure that you can see here, kind of has many layers. That's called the Golgi complex. The Golgi complex takes those proteins and packages them and sends them to their final destination. So if you have a protein that gets made that ends up in the plasma membrane, that's where it's going to get sent via the Golgi complex. If you have a protein that needs to go to the mitochondria, it's going to happen via the Golgi complex. You can see how it's kind of sending out proteins in these little pockets here to different areas of the cell. Other structures worth mentioning, hopefully everyone who's taken a basic biology class recognizes mitochondrium. Mitochondria are going to be the powerhouse of the cell, and they are going to make ATP, or adenosine triphosphate. It's going to be the energy source of the cell. So all of these processes that need energy to take place generally use ATP as the energy source. I'm not going to go through every single one of these just because I don't want to kind of overwhelm you. Something else that we'll talk about maybe is the cytoskeleton. That's going to be part of the internal structure of the cell underneath the plasma membrane that kind of gives the cell structure. So you can see some of these lines down here under the plasma membrane. Those lines are composed of all of these areas here, the microtubule, microfilament, intermediate filament, and those different filaments give the cell structure. These are going to be important when we talk about things like skeletal or muscle cell structure. So types of cells that have very specific structures or shapes to them, the cytoskeleton is going to be really important for giving those cells that type of shape. I'm not going to go again through each one of these. Things like flagellum and cilium, we're not seeing a ton in human cells. Sometimes we'll see them in eukaryotic or cells with a nucleus that are single-celled. Cilium we will see a little bit, maybe in epithelial tissue that helps kind of the movement of the flow of items through our intestinal system. So we will see some cilium there, but it's again, you won't see a ton of like flagellum located within the human body. Okay, so I'm just going to go through and quickly erase this. So I tried to circle kind of the main components that we'll be talking about. Don't feel like you need to have written down every single function of every single thing on this slide, because we're going to slowly but surely walk our way through the function of the most important cell components here. So this is just kind of a nice big overview, and we'll come back to this slide kind of at the end to just kind of capstone everything that we've talked about, but just make sure that you don't feel overwhelmed just looking at the slide going, oh my gosh, what is a nucleolus? Do we need to know what a glycogen granule is? No, not at this point, not from this slide. We'll slowly but surely kind of step our way through the different parts of the cell. Okay, ooh, I guess I didn't get everything erased. Okay, so the parts of the cell can be divided into three parts. So you have, first of all, your plasma membrane, which is, again, encompassing kind of all of the outside of the cell. It's a phospholipid bilayer. Bilayer meaning there's two phospholipids, and we'll look at that structure in the next slide. So that kind of encompasses and kind of keeps in everything on the outside. Next, within that, you have the cytoplasm. The cytoplasm is composed of two different things. You have both the cytosol and the organelles. So the cytosol is kind of the gelatinous goo that the liquid structure within the plasma membrane that kind of holds all of the organelles. It's kind of like allowing them to exist within that cell. The cytosol isn't just water. It's got a lot of ions, a lot of sugars, a lot of lipids that are contained within it. So it's not just a pure water environment. And then the organelles, as we talked about before, are going to be things like the nucleus, the mitochondria, the Golgi apparatus, the endoplasmic reticulum. Those are all examples of organelles that are contained within the cytoplasm, and each one of those organelles has a very specific function. And kind of like I said, cell's a little protein making machine, so many of those functions fall in line with protein production and getting those proteins to the right place so they can carry out other functions. So let's say if you had, let's say, a protein that needed to end up in the plasma membrane, the blueprint for that would be found in the DNA in the nucleus. Once that blueprint was read and made into mRNA, the mRNA would kind of travel outside of the nucleus and go to the endoplasmic reticulum, and the rough ER studded with ribosomes would make that into a protein. That protein would get folded and head over to the Golgi apparatus, and from the Golgi apparatus would get shuttled to the plasma membrane. So you do not need to memorize that whole process I just said, but I just wanted to kind of give it as an example. So if you had a receptor, let's say like an insulin receptor that needed to be expressed on the surface of a cell, that's exactly how kind of the steps of that process would happen in that protein factory kind of situation of the cell. So you've got the plasma membrane, the cytoplasm, and the nucleus. The nucleus is going to be comprised, have a couple different components in it. It's going to have chromosomes. So as a human, you have 23 chromosomes. If you ever have a hard time remembering it, always think of, oh, 23andMe. Why is it 23? Well, because there are 23 chromosomes that company sequences for you to give you your full genetic code or your genome. Within those chromosomes, if we looked at this, let's say a single chromosome, there would be specific genes, and genes are just kind of small subsections of an entire chromosome that code for a protein. So each gene codes for a very specific protein. And again, this is kind of oversimplified genetics talk here, but the nucleus does contain chromosomes, and within those chromosomes you have genes, and each one of those genes codes for a protein that has a function within your cells. So kind of just, if you'll notice a common theme here, it kind of keeps coming back to the how we get proteins made. But again, proteins are very much the workhorses of the cell and carry out a lot of the functions. As I'm giving you this lecture, and if you're looking at your own PowerPoint, recognize that there are these links that you can click on in your PowerPoint, and they'll lead to animations. So if you're looking at this slide and you've gone through it with me, and maybe you just need a little bit further explanation, feel free to click on those links within the slide. Okay, so today we're going to primarily focus on the plasma membrane. You might think of the plasma membrane as an area where there's not a whole lot going on, but really there's a bevy of activity constantly occurring at the plasma membrane, because it's really interacting with the outside environment, with the inside environment. And much like we talked about in chapter 1, maintaining homeostasis is very important for an organism, but homeostasis is very important to also maintain within a cell. And so the plasma membrane really helps mediate that homeostasis, or keeping the inside of the cell not having these huge fluctuations of, let's say, water fluxing in or leaving, or ions coming in or leaving. It's very selective about what it lets into and out of the cell. So it's a very important structure. We're going to talk about that today. So this is a great image just showing you the overall kind of general structure of the plasma membrane. Recognize that the plasma membrane is very fluid. It's made up of these phospholipids. So remember in chapter 2 we talked about phospholipids a little bit. There's that phosphate head that's going to have a charge on it, which makes it polar. So the outside of the phospholipid membrane is going to be polar. And again, if you remember, polar equals H2O loving. So it loves water, which means it's hydrophobic. philic. Inside the phospholipids here, you can see that there are lipids, which essentially lipids equals fat. And if if you've ever mixed oil and water, you can recognize that oil and water does not mix. And these lipids or fats, these lipid tails in the middle, are H2O hating, which makes them hydrophobic. They fear water. So hydrophilic are those water-loving heads. Hydrophobic are those water-hating, we'll go water hating tails. And so because of that, your body or your cells have a nice fat layer around them. And that fat layer essentially prevents any type of water-loving or polar molecule from passing right through it, because it's basically creating that little fat layer. So you've got an aqueous environment inside the cell, you have an aqueous environment or water area outside of the cell, and then that nice fat layer there. So if we look at the plasma membrane, you'll recognize here that there are a series of things that are actually embedded into the membrane or hanging out in the membrane. So we're going to talk about these a little bit more in the following slides. Things like glycolipids, glycoproteins, and if we look at the different kind of characterizations of the types of proteins that interact with the plasma membrane, you have integral or called transmembrane proteins, and you can see the 3 arrows here pointing to these transmembrane proteins. And if you look at these transmembrane proteins, hopefully you can start to recognize that they have a similar type of structure. They're all spanning the membrane. That's what makes them transmembrane proteins, is that they're all spanning the membrane, meaning transmembrane across the membrane. The other type of proteins are these peripheral proteins, proteins that don't go across the membrane. They're just associated with one side of the membrane or the other, so they can be associated with the inside of the cell membrane or the outside of the cell membrane. So those are both types of peripheral proteins. We're going to go into both of these far more in depth in the following slides. So integral crosses or transmembrane proteins cross the membrane. Peripheral proteins are just associated with either the inside or the outside, kind of decorating the inside or the outside, and are associated with other proteins. So I'm not going to go into any more kind of specificity there, because we're going to talk about it quite a bit in the following slides. So just make sure that you just take note of integral versus peripheral, and then kind of leave a bunch of space below that, because we're going to talk about them more and what types of proteins are in those categories. Okay, so this next slide, you have got integral, which are also called transmembrane proteins. So the example that you can see here, one is this glycoprotein. This glycoprotein spans the membrane. You can see that it's got this kind of curlicue type of structure here that's kind of representing an alpha helix. It's a glycoprotein. If you remember, glyco kind of looks like glucose, which means it's basically a protein that has this sugary, substance that's attached to it. Now that sugary substance, the cells often use sugars as kind of hallmarks or tags to tell you what types of cells they are. So a great example of that is immune cells. Immune cells are great at using sugars as tags to tell the body self from non-self. And again, that's one of the major functions of the immune system, is to discern self from non-self. And so these sugary tags that are associated with these proteins can really help the body do that. The next type of integral protein that we'll see here is a pore. I'm sorry, the pore is the area between it, but this channel protein is the next type of protein we'll talk about. And this channel is basically creating this pore, which allows selective passage of different ions or molecules into or out of the cell. So this pore that you see here is what allows the movement. It's the space in the center of the channel protein that allows passage through the channel protein into or out of the cell. So like I said, that's important for the cell to maintain homeostasis. So in order to do that, it has to be very selective about what it lets in and what it lets out. So in order to let things in and out, it has to form kind of these channels or these little like doorways for the ion or molecule to pass through. So channel proteins are the way that that happens. So the last type of integral membrane protein or transmembrane protein that we're going to talk about here is this little guy here. And it's interesting, it's not labeled, but it's a receptor, RECEPT. TOR. So the importance of receptors are the fact that they transduce information from the outside of the cell to the inside of the cell without necessarily letting anything pass through it. So in this case, if you have something, a molecule that binds to this receptor, what it's going to do is cause this receptor to change shape, and this receptor is going to send information to the inside of the cell saying, hey, the cell needs to respond in this way. So an insulin receptor would be an example of a receptor. Any receptor that binds a neurotransmitter, like a serotonin receptor, would be a type of receptor. And so those types of molecules don't necessarily come into the cell. What they do is they bind to the receptor, and the receptor tells the inside of the cell, hey, we need to respond in some way, something needs to change. So the receptor is kind of the third kind of integral transmembrane protein. And second, we talked a little bit about these peripheral proteins that you can see here. Peripheral proteins are just associated with the inside or the outside of the plasma membrane. Often those are structural proteins that help the cell kind of stay in its proper shape. So a great example of a peripheral protein would be something like actin. Actin creates structure inside the cell and gives that cell the shape that it needs in order to carry out its function. Even when we're talking about things at the cellular level, structure function is important. And as we go through this semester and we talk about some of the larger tissues and organ systems, structure function relationships are going to be even more important when we get there. Okay, so we're talking about these different types of membrane proteins that are embedded in the cell surface. So they can serve a variety of functions. So this over here is just kind of a nice laundry list of the different functions that membrane proteins serve. So we mentioned that there's an ability for a channel protein to be an ion channel. An ion channel allows the passage of ions into and out of the cell. Popular ions that pass through that ion channel would be ions like sodium, potassium, chloride would all be examples of ions that would pass into and out of an ion channel. This is an integral membrane protein, meaning it's transmembrane and passes through each side, has access to both sides. Next, we can see a carrier protein. A carrier protein transports a molecule from inside to outside the cell, but you can see here in the channel it's either open or closed, and with the carrier protein that we see here there's a conformational change or a change in the structure of the protein. So at one point it's open to the outside of the cell. Once it binds to that molecule of interest, then it changes shape and it releases the molecule to the inside of the cell. So the example that we'll talk about a little bit later is glucose. Glucose is often brought into the cell via a carrier protein, and this is a carrier protein that lines all of your endothelial cells of your intestines. Next, like I said, we have the receptor. Receptors recognize specific, we call ligands, or these are just molecules that bind to the receptor, telling the receptor to activate. And so an example, the antidiuretic hormone binds to receptors to kidneys and changes the permeability of water. So in this case, you've got this you have a hormone that's binding to this receptor, telling the inside of the cell to change water permeability. So that's one example of a receptor, but there are many examples of receptors that we'll talk about over the course of the semester. Another function of membrane proteins would be enzymatic function. Enzymes, if you remember from earlier lectures, we talked about enzymes and how they lower activation energy or they catalyze reactions. And so there are some reactions that are really important to happen right there at the cell membrane. So example is the lactase retreating from epithelial cells lining the small intestine splits the disaccharide lactose in the milk you drink. So this is an example of a way that the cells that line your epithelial cells are breaking down the food that you consume, things like milk, and the enzymatic function, breaking down those molecules into smaller molecules for your body to be able to process them. So anytime you see a molecule being broken down, it's usually by some type of enzymatic function, if not by maybe a change in pH like we see in the stomach. Okay, so enzymatic, linker, integral, and peripheral, these are just anchors and filaments inside the outside the plasma membrane providing structure. So linker proteins are going to be providing the structures of the cells themselves. And then the last one that we have down here is a cell identity marker, a glycoprotein. Like I said, these proteins are going to span the surface of the phospholipid bilayer, and then you're going to have the glycos or the sugars that decorate the surface. A great example here is the MHC protein, which stands for major histocompatibility complex. You do not need to memorize that. Just recognize that these glycoproteins can be utilized by the immune system, like the major histocompatibility complex, which again allows the cells to identify self from non-self. So those glycos are sugars on the surface of the cells, allows the body to recognize self from non-self, which is a major function of the immune system. Okay, so for membrane fluidity, so membranes are very fluid structures. They are not like the cell walls that we see within plant cells. They're far, more fluid, and can actually have a lot of different shapes and structures. If you think of the structure of a nerve cell versus the structure of an epithelial cell, their structures are very different. However, their basic components are the same in how those structures are kind of acquired. So just recognize that the membrane itself is very fluid, and within that membrane, plasma membrane, you can have receptors, or all of these integral proteins that we're talking about kind of move freely within that. It's almost like if you were standing in a ball pit and you and a friend were, way far away from one another, then you decided to like walk towards one another. That's kind of like how receptors and proteins can kind of move through the plasma membrane. It's not a super easy movement, but it can kind of like move slowly but surely through. Now, if you want to talk more about membrane fluidity, and there are current theories about how there are certain structures that kind of stick together in the fluid. It's called a lipid raft, but it's far outside of the scope of this course. So membrane lipids and proteins are mobile on their own half of the membrane. So if a lipid or a protein is on the, let's say, inside of the cell or intracellular side, or on the outside of the cell or the extracellular side, it can only stay on that side. It's very difficult for them to flip from one to another. There are some examples of proteins that can do that, but we won't talk about that specifically in this course. Something else to mention here is that cholesterol serves to stabilize the membrane and reduce membrane fluidity. So different levels of cholesterol can actually alter the way that the membrane itself interacts with its ability to be fluid. Okay, but plasma membranes are selectively permeable, and again, that's one of kind of the major functions of the plasma membrane, is to be selective. So the lipid bilayer is always permeable to small, non-polar, uncharged molecules. So if we think about that plasma membrane, right, you've got that fat, those lipids on the insides that are water hating and are non-polar because they're water hating. Non-polar, well, they're non-polar, which makes them water hating. Non-polar, and then on the outside of the cell you have those phosphate heads that are water loving and polar. So you've got that nice fatty layer around the cell. So in order for something to pass through the cell, it has to be, again, very, very small. So it could just be like a small little dot and sneak its way through. It can be non-polar. So non-polar, the reason it can get through the cell is because the membrane has those lipids that are also non-polar. So it's like, hey, you're non-polar, I'm non-polar too. I can sneak my way through here. And then uncharged molecules. And so because this is non-polar, uncharged molecules can also make their way through that non-polar. So if it's too small, non-polar, uncharged molecules, those are the types of molecules that are most likely going to make it through that plasma membrane. So because the cell sometimes needs larger molecules, polar molecules, or charged molecules inside the cell's kind of way to have that happen is to have channels or transporters that kind of increase the permeability of the cell. So we've talked about that a couple of different times, how there are these proteins that are embedded into the surface of the plasma membrane that act as little doorways for these larger molecules to get into. Macromolecules are only able to pass through the plasma membrane by vesicular transport, and we haven't even kind of introduced what vesicular transport is. But if we look at the cell again has that nice large plasma membrane. And what vesicles are kind of small little blebs within the um, within the cell that allows the cell to either send things out of the cell or to bring things into the cell. So let's say you had a macromolecule that wanted in, it would come and bind to the cell surface, and then that cell surface would start to divot in, and then that would almost engulf that entire macromolecule. So if we looked at the next kind of vesicular step of vesicular transport, that bleb, this is the next step, that bleb would be engulfed entirely. So inside would be that macromolecule, and it would still have parts of the plasma membrane around it. And again, that's what a vesicle is. And this is a very quick description of what vesicular transport is, but the plasma membrane essentially has that fatty layer, and something binds to it. It starts to engulf it slowly and almost create a sub little plasma membrane inside, and that's a vesicle. And then if that macromolecule needs to be released into the cell, that's kind of the next step in vesicular transport. So again, that's, I think, a little bit too specific for what we're talking about in this class. We'll focus primarily on these other types of transport, things that are mediated through channels and transporters. Okay, so how do things kind of get forced into and out of the cell? Like, how does the cell decide, like, what comes in, what goes out? Why does it get kind of forced into or out of the cell? And one of the ways that let's say ions or materials kind of decide that they need to kind of go inside the cell is because there's this concentration gradient, or there's this difference in the concentration of a chemical on one side of the plasma membrane or the other. So if we kind of, again, I'm going to draw it down here. If we have the plasma membrane, and we're talking about a difference in concentration, probably the easiest example would be sodium ions. So especially for nerve cells, there's a lot of sodium ions outside the cell. We'll go Na+. That's what those pluses mean. Na+. There's lots of sodium outside the cell and very few sodium inside the cell. So this concentration gradient basically states that something of high concentration wants to go to an area of low concentration. So because of this, sodium wants to force its way into the cell. Now, so lots of sodium outside the cell, little sodium inside the cell. The sodium wants to come into the cell. Now that's not the only gradient at play that's driving sodium into the cell. So the other kind of ion that's also playing is you do have negatively charged ions inside the cell as well. And if we look at the charge of like a nerve cell, the overall charge of a nerve cell is negative on the inside. So you have basically what's generated here is an electrical gradient. It's the difference in concentration of ions between one side of the plasma membrane and the other. And this these ions are causing a difference in charge. And so not only do you have a concentration difference of sodium, but you also have a charge difference between the inside and the outside of the cell. So these positively charged ions that you see here are attracted to coming inside the cell because of the internal negative charge of the cell. So the negative inside of the cell because of this has a electrical gradient, and we'll talk about this when we talk about nerve cells. If you want to put a number to it, it's can be -50 for some cells, and this negative number essentially attracts this positively charged sodium ion to the inside of the cell. So this is essentially what kind of establishes what's called the electrochemical gradient that's so important for nerve cells to function. So there's not only a difference in concentration between the inside and the outside of the cell of these charged molecules, but there's also an electrical difference that's giving rise to it between the inside and the outside of the cell. And we'll talk far more about the electrical chemical gradient when we get to talking about the nervous system. Okay. So we've talked about the electrochemical gradient, about some of the proteins that are embedded in the cell. Let's talk about how molecules specifically get inside and outside of the cell, aka how do they get transported across the plasma membrane. Okay, so there are a few different types of processes that allow for movement across the cell membrane. First, you have passive processes, things like simple diffusion, facilitated diffusion, and osmosis. We'll talk about these passive processes. They're very dependent upon things like concentration, and the movement of things from an area of high concentration to low concentration. Now, I keep using those terms, and maybe that doesn't make sense to you, but think about if you've ever gone to a club and you are inside the club and it's super packed and it's hot, and you're like, I want to go from an area of high concentration of people to an area of low concentration of people, which is outside. So there are many ions that kind of have that same like idea, like I need to get away from this very, busy environment and go to an area of far fewer other molecules. And so I'll probably continue to use that kind of club metaphor throughout the rest of this lecture. So that's kind of how passive processes are based off of concentration, moving from an area of high concentration to low. Active processes require a level of primary, secondary transport or vesicular transport. We'll talk more about those. Active processes are going to require energy. And so they're going to require energy or also utilize a secondary type of transporter process. So we'll talk about some of those active processes when we get to that point. Okay, so here's another opportunity for you, again, if you are maybe needing more of a visual representation of what I'm describing, please feel free to open up your own PowerPoint and click onto these particular links. I would do it now, but the Zoom doesn't actually follow me to those links, and so I can't really show you the video. That's how I ended up with a dead spot in that first lecture. Okay, so first let's talk about passive transport and simple diffusion. So things that are going to influence diffusion are going to be things like the steepness of the concentration gradient. So if you have a very, very high concentration, and this is represented by blue substance at the bottom of this beaker, beaker, you can see that there's a high concentration of blue substance here and not basically none at the top. And so kind of the darker that this area is down here, probably the worst color possible. The darker this color is down here, the faster this diffusion would occur, because you would have a much higher concentration located at the one end. Things like temperature can also influence diffusion. You can think of temperature as being kind of a representation of kind of kinetic energy. So the higher temperature is, the more kinetic energy, meaning the more the molecules are moving, the faster the molecules are moving. So the faster the molecules are moving, not surprisingly, the faster they would be diffusing from one area to another. So it's almost like the same analogy when we're talking about you being in the club. It's if you were diffusing from the inside of the club to the outside of the club, it'd be the difference between you slowly walking out there or you basically running out there. That's going to influence the rate that you diffuse to the outside. The mass of the diffusion substance also can influence the rate of diffusion. So how large that is in terms of its mass is going to move across that aqueous environment. The surface area of that substance, and then the diffusion distance. So again, if you are, let's say, leaving the club, which is maybe a small club, or and you just can walk right out the door, versus let's say you were at the Grizz Stadium and you wanted to leave a football game, probably would take you a lot longer to get out of the stadium and to diffuse from the large crowd because of that. So that diffusion distance is greater, much greater there. So again, all of these things can influence the rate of diffusion, and the cell uses this quite frequently. So facilitated diffusion is slightly different because you have proteins that are going to assist in the movement of proteins are going to assist in the movements of these molecules. And so they can help solutes that are two polar. Again, two polar, which are water loving and going to have a hard time getting across that fatty kind of layer of the cell, or two charged. Again, charged is going to make something polar to move through this lipid bilayer. Again, that fatty lipid bilayer is very nonpolar and doesn't really like polar substances, and doesn't really like large molecules or charged molecules. So you can have either channel or carrier-mediated facilitated diffusion, and we're going to talk more about the channel-mediated and carrier-mediated facilitated diffusion in the following slides, so I'm not going to go into it a ton here. Okay, so similar to what we had seen with diffusion, I think this is a really good slide just showing you how diffusion occurs, and again, what's driving it. It's that difference in concentration gradient. So you've got a concentration gradient of a lot of molecules outside the cell and very few of those molecules inside the cells. So the driving force, what they call driving force, for these molecules would be to go inside the cell, away from the highly concentrated areas. And so that can occur through simple diffusion. Simple diffusion is only going to occur with the small molecules, the nonpolar molecules that we talked about earlier that are able to kind of slip by all of those nonpolar lipids, and then the uncharged molecules. So those are going to be the molecules that are able to actually undergo simple uncharged charged. Going to be the molecules that are able to undergo simple diffusion. Now, molecules that are much larger or are charged are going to have to use another protein for that to occur. And so channel mediated is another type of diffusion. So in this case, a channel, sometimes a much larger molecule, let's say sodium, sometimes potassium are examples of these channel mediated. And that's basically channel mediated can be an open and closed situation where you have the channel open and then you have it closed. Next, we'll talk about carrier-mediated facilitated diffusion. Here you can see a molecule that looks far more complex. This is actually a sugar. We'll talk about, I think it's glucose, so sugar, the sugar and the channel-mediated facilitated diffusion we'll talk more about in subsequent slides, but you have kind of this conformation that's open to one side of the cell. You have the sugar or the molecule bind, and then it's going to change conformation, and that molecule ends up on the inside of the cell. So there's much more I say conformational change, and it's basically just kind of a shape change of the protein that allows it into the cell. So that shape change of the protein that spans the plasma membrane. So the carrier protein there that spans the membrane is going to undergo a shape change to allow that molecule of glucose into the cell. So channel-mediated facilitated diffusion, we can see here that the example that's given is of a potassium channel. So this potassium channel is interesting because it has a pore. The pore allows the passage of that positively charged ion across the plasma membrane. And you can see that this particular channel is kind of a little different because it also has a gate. We'll talk about that gate a little bit later on when we're talking about the central nervous system. I don't necessarily think it's super important right now, but I guess it's worth just noting that for a channel, I talk about it being a super easy open and closed status, but there can also be instances where gates do occur, and those gates kind of just plug up the hole and allow that channel to not be passed through anymore. And we'll talk about what actually mediates that channel being open and closed when we get to talking about the central nervous system. I think we've got plenty of other parts of the topic to talk about here. Okay, so that's channel-mediated facilitated diffusion. In this case, we're looking at carrier-mediated facilitated diffusion. So you can see here glucose, probably in the gut. These are epithelial cells, possibly binding to the carrier-mediated, the carriers that are going to bind to glucose, and then there's going to be a change or a shift in the shape of the protein, the integral membrane protein, that allows glucose to be released to the inside. So it's open to the outside, and it goes through that shape change, and then it gets open to the inside. So versus a pore, which is just kind of like open and closed like this, the carrier mediated kind of has this open and closed state towards the inside and the outside of the cell. We can see here, similar to what we saw on the last slide, is that this is concentration driven. So there's much more glucose outside the cell versus inside the cell. And so that's what's driving, essentially giving the cell the ability to move this from outside to inside. Okay. Okay, so we've talked about carrier-mediated, channel-mediated, and now we're going to talk about passive transport via osmosis. So osmosis is basically the movement of things like movement of water from one side of a membrane to another based off of a selective membrane. So the net movement of a solvent through a selective permeable membrane from an area of high concentration to an area of low concentration. So we can think of this, we basically have a tube here filled with water, and there's water on one side, and there's another side that has water plus a solute molecule. Now you'll recognize that the solute molecule is only located on one side, and there's a selectively permeable membrane that is not allowing the solute molecule to go from this side to this side. So that does not happen. That solute molecule can only stay on the one side of that plasma membrane. So what happens here is, because of that higher concentration of the solute molecule on one side, what happens is you have water that's basically moving from the one side to the other to try to essentially decrease the concentration of the solute molecule. Because what essentially the cell, or in the case of a selectively permeable membrane, wants to happen is you would like to have equal concentration of the solute on both sides. But since that isn't possible, what happens is you've got movement of the water from one side, the highest side of low concentration, to the side of high concentration of solute. It's trying to essentially kind of dilute out that solute, so it would have the same concentration on both sides. So the movement is due to hydrostatic pressure. Now, what also can occur is you can apply pressure or osmotic pressure to one side, and that pressure again requires energy. So you've got to put some type of force down in order to kind of reestablish that equal amounts of volume on both sides, inside and outside of the cell. But again, that requires some type of outside pressure to occur. But if you have a higher concentration of solutes on one side, water wants to move in. So hopefully you can start to kind of see how this would apply to cells. So if you had a cell that had a higher concentration of solutes inside the cell, the water would want to move inside the cell to try to essentially to dilute out that solute, so there would be equal concentrations on both sides. So equal concentrations is what the cell is always trying to get to, so it uses it through movement of water, and then in some cases it moves the ions themselves. So in this case we're talking about the movement of the aqueous, or the solvent. In the case of cells, we're talking about water. So this influences the tonicity of cells. It's the tonicity of a solution relates to how the solution influences the shape of body cells. So if we look at the case here, we can have what's called a isotonic solution. Isotonic means essentially there's the same concentration outside the cell as inside the cell. And red's probably the worst color to use here. So this concentration is the same. They equal each other inside the cell and outside the cell. Because of that, you've got the equal movement of water into and outside the cell. So the example that we're looking at here is a red blood cell, and because it's an isotonic solution, meaning it's got the same concentration of ions, that's going to allow for the equal exchange of blood cells. So if you've ever worked, let's say, in a lab and you're dealing with biological substances, you'll recognize that the concentration of ions and things like that can influence the health of your cells. And so that's why things like saline are used instead of just straight up water when we're giving rehydrating individuals. Is you want the concentration of ions to be the same and not disrupt the cells. And we'll talk about kind of what happens when you do that. So this is a isotonic solution where you have the same concentration outside and inside the cell of solutes. Next, you can have the hypotonic solution. In the hypotonic solution, you have a much greater number of solutes inside the cell, far fewer, sorry, that's a concentration sign, outside the cell, and because of that, the water wants to flow into the cell, and in a hypotonic solution, water flows into the cell and can lead to lysis or disruption or bursting of the cell. Now, in the case of a hypertonic solution, you have a high concentration. Wow, these colors are not great. You have a high concentration outside the cell and a low concentration of solutes inside the cell. And because of that, water wants to leave the cell, so it's leaving the cell. And then you have more water leaving the cell than going in. So what that leads to is prenation, or kind of like a raisin type structure, dehydration types of structure. And that's the case of a hypertonic solution. So I guess in terms of the way that I've kind of remembered this hypertonic. Hyper, I think of a lot of things going on, being very excited. Think of that as the external, like there's a high concentration going on outside the cell. It's hyper outside of the cell, so water flows out. Hypotonic is just the opposite. You've got not that much going on outside the cell, so the flow water wants to go in, so you're going to lead to cell bursting. And then isotonic, again, is that nice equal exchange both into and outside of the cell, leading to kind of a conservation of the cell shape. So you can see here how tonicity can influence cell shape based on the flow of essentially water into or outside of the cell, based on the concentration of the solutes. Okay. Oops. Okay, so this particular image is just showing you active transport. Ooh, this is not my favorite. I wonder if I can select this. Okay, just bear with me for a second. I want to gonna exit out of presentation mode here, just because I think this is making it look very confusing. Okay. So we talked about the different types of passive transport, and next what we're going to talk about is different types of active transport. So active transport is different than passive because it requires some type of input of energy. So energy derived from ATP changes the shape of a transporter protein, which pumps the substance across the plasma membrane. So in this case, we're looking at examples of the use of ATP or energy of cells. So if you remember earlier, we talked about kind of the concentration of sodium. Sodium wants to go into the cell because there's a high concentration outside of the cell, and potassium has a high concentration inside the cell and wants to go out of the cell. So this is kind of the example that we're talking about here is a nerve cell. I always think of the nerve cell as a salty banana because bananas have a ton of potassium, and they're salty, meaning that there's sodium on the outside. So a salty banana, well, I'm using it here. We'll use it again when we get to talking about the central nervous system. So you can see the concentration gradient of sodium wanting to come into the cell and potassium wanting to leave the cell. So the sodium-potassium pump is a great example of active transport because what it does is it helps maintain this balance of high concentration of sodium outside the cell and high concentration of sodium inside the cell. So even though there is a low concentration of sodium inside the cell, what it does is it binds to that sodium and pumps it out of the cell. So you'll recognize here that it requires energy to pump things against their concentration gradient. So if it's got a high concentration outside the cell and a low concentration inside the cell, in order for this movement to go out, it's going to require energy. So in this case, we're using ATP to cause this, let's say in this case, to go from a low concentration to a high concentration. So to move it against its concentration gradient, you're going to require energy. So if you want to move something from where there's a low amount to where there's a high amount, you're going to require energy. It's almost, again, like if we go back to the club scenario, if you're wanting to leave the club, but the bouncer says, no, we got to keep our numbers high, he's going to use his forceful energy to keep you inside the club and move you from that area of low concentration to high concentration of people inside the club. Again, maybe that analogy works for you, maybe it doesn't. If you hate the club, I apologize, but you know, it just hopefully gives you a nice visual. So in the case of the ATP, the use of ATP, here you can see ATP being broken down from ATP to ADP. We'll talk about that a little bit later. But similarly, as you're moving sodium outside the cell, you're moving potassium into the cell, and again, this is against its concentration gradient. So the sodium-potassium pump does this. It kind of moves these two molecules against their concentration gradients, and because it's against their concentration gradients, it requires energy. So it's a great example of active transport. So the sodium-potassium pump exists in our neurons, and we'll talk about it far more when we get to that chapter, but it's a great example of active transport. Other ways in which active transport can take place is that you can use concentration gradients to move ions across it. So it doesn't necessarily have to be energy in the form of ATP. You can use other concentration gradients. So in the case of, let's say, this sodium, remember sodium has a high concentration outside the cell and a low concentration inside the cell, so the cell wants to bring sodium in. So what this particular transporter has done is it's coupled that with the movement of calcium, and it's basically using this transporter to move calcium outside the cell against its concentration gradient. So when I say against, it's from an area of low concentration to an area of high concentration. And so in order for that to happen, there has to be some type of energy. And in this case, we're using the energy that's established through the concentration gradient of sodium. So for sodium, you're going high to low. So it's using that movement here of high to low. So high to low, and then the calcium is being binding here and moving from low to high. So because it's moving from low to high, the only way it can actually go through that different conformational change in this transporter to work is it's coupled with that sodium. So it basically hijacks that concentration gradient that sodium uses to move itself from an area of low to high concentration. And it's important that the cell keep calcium very low, concentration of calcium very slow inside the cell, because it's a signaling molecule. Specifically, when we talk about muscle function, you'll recognize how important calcium is. And so that's an example of why this transporter is important, kind of at the larger picture. The same thing can be said for the hydrogen ions that you can see here, the same kind of coupling with the sodium transporter. And then things like glucose and amino acids, again, are still using that sodium gradient in order to kind of move things with or against that concentration gradient. So you're not necessarily always moving in opposite directions. You can have them moving in the same direction. So in the case of glucose, let's say you have a low concentration outside the cell and a high concentration inside the cell, you can still use that sodium gradient to get glucose into the cell. Same thing can be said for different amino acids. So again, the big kind of takeaway from this particular image is that you can use sodium or any type of concentration gradient to drive other substances against their own concentration gradient. So when I say against, I mean you're making them go from an area of high concentration to low concentration. So just remember, things want to go from the very, very busy packed area to the very less packed area. So when you force them to go the other way, they're not very happy about it. So it requires some type of energy. Okay, the last type of movement that we'll talk about today, transport, I kind of alluded to earlier in the class, is the receptor-mediated endocytosis through vesicles. And so what happens here is if you have a substance that binds to the surface of the cell, what's going to end up happening is the cell's basically going to engulf that item. And so when I say that, it's almost exactly what it sounds like. Engulf kind of meaning like it's almost like Pac-Manning in that receptor, whatever that receptor has bound. So you've got all these receptors here, receptor binding LDL. Once it's bound to that, it's going to start to Pac-Man in that particular receptor with all of those things bound to it. That vesicle is going to form. And it's got all of these kind of spikes on the outside of it. And then those spikes can be lost, and those are called clathrin, but those can be lost, and then it can fuse with what's called an endosome. And then basically kind of the downstream effect of this is there's two things that can happen. Either this vesicle can be recycled and sent back up to the cell surface to do its job again, or it can get degraded and kind of broken down. I really do feel like vesicle and receptor mediated endocytosis is kind of outside of the scope of this class. So basically this slide is just kind of for your information. You will not be tested on it, just because I think, again, I don't think we necessarily need to be going into the depths of vesicle mediated endocytosis. Just recognize that it exists. You can have this thing that binds to a receptor, that receptor gets engulfed into the cell, and the cell has different things that it can kind of do with it. But I don't need you to understand what clathrin is. I just think, again, that's outside of the scope of this course. Okay, other ways in which active transports can occur is vesicles through phagocytosis. This occurs in microbes far more than we see in the cells within our body, with the exception of maybe some of our immune cells. We do have white blood cells that do engulf, let's say, a yeast cell. So that's kind of the one exception that we see phagocytosis occur within the cell body. And it's basically almost like I said with the last one, where you've got this kind of Pac-Manning of this thing. So you've got this this foreign body that the white blood cell kind of identifies, and it goes in and literally just kind of eats it and consumes it, brings it into what's called a phagosome. And in the phagosome, you can see here, basically that thing gets kind of degraded. So it's almost like cellular eating. It's a way that cells kind of eat objects. And so it's very, if you ever watch a video of it, it's actually pretty cool to watch a single-celled organism really just Pac-Man this other thing and then consume it, bring it into its own body, and then degrade it. This is, again, a very popular way for single-celled organisms to consume things, but we do utilize it via white blood cells if it does identify a foreign body. Okay. So pinocytosis is very similar to phagocytosis, with the exception of pinocytosis is not for a specific, let's say, object. Pinocytosis is kind of like cell drinking. It's actually when the cell just uptakes water or some type of aqueous fluid. So pinocytosis is kind of the same thing, except for you just have cellular drinking that occurs. So formation of vesicle, fluid dissolved in the solutes, the lysosome and the digestive enzymes break that down, and digested solutes get released. But again, you're just kind of taking in, it's like kind of gulping in its environmental substance, but it's not necessarily, again, breaking things, pulling in larger objects. It's just the solute solution around it. Okay, exocytosis and transcytosis. Okay, so exocytosis, so we were just talking about different types of kind of vesicles coming into the cell, and vesicles can also leave the cell. So endocytosis is basically what we just talked about, endo, E-N-D-O. Endocytosis is basically coming into the cell, and then exocytosis is going out of the cell. And so exocytosis, most often when you have membrane enclosed vesicles fuse with the plasma membrane and release their contents into the extracellular fluid. So endo bringing in, exo going out. So that's going to be really important when we're talking about secretion of different enzymes, secretion of different fluids. And so you can think about maybe the oils that are secreted by your integumentary system that keep your, your body nice and kind of not completely dry. And then we can think about some of the secretory enzymes that kind of get released within our digestive system that allow for the breakdown of the molecules that we consume. So there are a couple of different places where we'll talk about exocytosis in the coming chapters. So we're going to be releasing things from inside the cell. That's one way to release large amounts of let's say proteins or enzymes that are created by the cell into its environment to carry out the functions. Transcytosis is in a combination of endo and exocytosis used to move substances from one side of the cell across it. And we'll talk about some types of transcytosis, specifically when we're talking about cells that have different sides. So if we have a cell has two different sides, we'll say like a basal and an apical side, which does exist. The cell's not completely the same all the way around. It's got different sides to it. We'll talk about how it moves things from one side to the other and why that movement is important. So I'm not going to torture you with each kind of step of these processes, but just recognize that when we get to talking about some of the cells that make up different systems, all these terms will be important. Okay, so We'll just do a quick kind of comparison of the different types of transport types. So passive processes, you're moving things down the concentration gradient. So we're going from an area of high concentration to an area of low concentration. It does not require ATP because you are using the concentration gradient. Diffusion, down a concentration gradient again, so it's very similar from an area of low to high. Due to their kinetic energy, until they reach equilibrium, they're hoping to kind of reach a place where that concentration is the same on both sides. In simple diffusion, very similarly, passive movement down its concentration through the lipid bilayer. The substances transported are going to be those nonpolar hydrophobic oxygen, carbon dioxide. Those are all examples of substances that will go through simple diffusion. Facilitated diffusion is going to require the placement of larger proteins, so through the lipid by transmembrane proteins, and function as channels or carriers. So facilitated diffusion is going to require integral membrane proteins. You can move much larger substances, charged substances, polar substances, using facilitated diffusion. So the examples that we talked about earlier were potassium, chloride, sodium, and calcium. Those charged molecules are only able to cross that lipid bilayer through facilitated diffusion. So they're going to require those integral membrane proteins as kind of doorways to the inside of the cell. Okay, we talked about osmosis and the movement of the water across the selective membrane from an area of higher to lower concentration until equilibrium is reached, and that transports the water in living systems. Active processes, again, probably the big thing to remember about active processes require energy of some form. Most often the energy that we talk about here is ATP, but it can be the energy that's acquired through concentration gradients, which is kind of considered to be potential energy. So active transport, we can move polar or charged molecules against its concentration gradient by transmembrane proteins. The sodium-potassium pump was the first example we gave of active transport, using ATP to move both sodium and potassium against their concentration gradients. So primary active processes, again, the sodium-potassium pump that we just talked about, uses the ATP and can move things like sodium, potassium, calcium, hydrogen, iodine, chlorine, and other ions against its concentration gradient. Secondary active transport, this is different because it's coupled. So you have it coupled to a concentration gradient. So in one side, you've got that concentration going from high to low, and that's the concentration gradient that you're using, but that's coupled with a movement, let's say, of something going from an area of low concentration to high. So the only way that that can happen is through the co-coupling of this other concentration gradient. So these are considered antiports, meaning going in opposite directions. And then symports would be two concentrations that were moving in the same direction, both into or both out of the cell. So those are all considered secondary active transport processes. Okay, and lastly, we talked about transport in vesicles, moving these large substances into or out of the cell. And we talked about endocytosis, receptor-mediated endocytosis. Again, vesicular transport, I don't think we necessarily need to go through like every step of the process in depth, but just recognize that something binds to a receptor, it gets brought into the cell, into its own little vesicle, and that vesicle can either be degraded or it can be recycled back up to the surface of the cell. Phagocytosis, cell eating. Again, that's how our white blood cells are going to consume things like bacteria and viruses. And bulk phase endocytosis, solutes and extracellular fluids, or pinocytosis is also what it's called. And then exocytosis, it's where you're bringing something from outside the cell inside the cell to outside of the cell. So endocytosis is in, exocytosis is out. Endocytosis is in, exocytosis is out. So exocytose, you're going to be releasing larger molecules, things like neurotransmitters, hormones, and digestive enzymes are all examples of exocytosis. So we will be talking about these processes a little bit later, but just again, I don't want to get too far into the weeds here. Transcytosis is when you have something that's brought into the cell on one side, travels all the way through, and gets released onto the other side. So antibodies that cross endothelial cells is an example. And this is just a common route substance between blood plasma and interstitial fluid. So you're moving from one side to the other using transcytosis. Okay, so again, we're kind of coming back here to the structures of the cell. Now, interestingly, we just spent an entire lecture just talking about this plasma membrane, right? And you're just like, wow, I can't believe we just spent an entire lecture talking about that, but it's very important because it's kind of the gateway into and out of the cell. So how that gateway is kept in the way that cells do that is important. Sometimes that gateway is utilized just to bring things into the cell needs to function, but in some ways it can actually drive the cell's function itself. So the movement of like sodium and potassium ions actually are what allow nerve cells to send signals. So that plasma membrane, there's a lot going on there, as you can see from this lecture. So just make sure you kind of step through and really think about what's moving where. I would say kind of the big picture is think about what type of molecule is crossing the plasma membrane, its properties, whether it's polar or nonpolar. Again, you'll be like, why are we learning chemistry in chapter 2? And it's like, for this reason. If it's polar or nonpolar, that's going to dictate whether it can passively cross the membrane or whether it needs some type of carrier or channel. And then also think about with carriers and channels, also always be thinking about concentrations. Where's the concentration of the substance higher and lower? So probably the simplest kind of concentration that I'll continually throw out there is the cells of salty banana. High concentration of sodium outside, high concentration of potassium inside. There's also low concentrations of calcium inside, high concentrations of calcium outside. And so just kind of recognizing that. But we'll probably use sodium most frequently as the example, just because I kind of keep talking about it. We talked about many ways in which its transport was used to move other molecules. So passive and active transport processes, as well as the endocytosis processes as well, another way in which we are able to get things into and out of the cell. Okay, so the cytoplasm, kind of moving further into the cell just outside of the plasma membrane, has two components. You've got this cytosol, which is known as intercellular fluid portion of the cytoplasm. Given what we know about osmosis, recognize that it's going to be composed of solutes and ions and proteins. That's going to change kind of the concentration of cytosol. And then you also have organelles that are specialized structures that have very specific functions within the cell as well. So we're going to talk more about these organelles, each one of those organelles, and kind of what their functions are in the next lecture, so please stay tuned. The last thing I just do want to do is remind you is that the answer study guide questions for chapter 3 for Tuesday are going to be taken from this lecture, so just come prepared to discuss those. And then quiz 3 is due by Friday at midnight, so make sure you've completed that. And then homework 4 for the adaptive practice for chapter 4 is due the following Monday. After you've watched this lecture, if you have questions or concerns, please reach out. I'm happy to clarify some of the concepts, and thank you for your attention.